Hi, my name is Brianne, and I'm a graduate of the University of Windsor, where I completed both my Bachelor's of Science degree and my PhD in biology. I now work in the field of cancer research at the university as a research associate and in the chair of our RIOT or Research Information Outreach team, working to bring information about cancer research to the community. Today, you'll learn about some cancer basics and how researchers work together to learn more about cancer and find new ways to treat it. Then you get to test your knowledge of Cancer 101 and try your hand as a cancer researcher and learn more about the ins and outs and behind the scenes of the cancer research. Now, cancer isn't just one disease, it's many different diseases. And even within all these different types of cancers, at a very basic level, cancer is characterized by uncontrolled cell growth caused by a genetic change that ultimately affects the function of a cell. Now, normally, all of the tissues in our body are very organized. When you look at cells in these different tissues within our body, whether it's our skin, our lungs, or liver, or bladder, no matter how different they may look, what you'll see is that there's a pattern to how these cells are arranged. And our cells are very smart and normally know exactly how to keep things just right and the exact moment they might need to grow and divide to replace old cells. Sometimes, though, a single cell might get a genetic change. Now, this may not do anything, and it could be completely harmless. You may not even notice it. But sometimes, this genetic change allows the cell to begin to grow unchecked. If this continues over time, you'll end up with essentially a pile of cells, and when you look at this under the microscope, it will start to look much more disorganized. That normal order you see in the tissue is now gone, and the cells are growing wherever they want. So what causes these genetic changes in the first place? So there are a lot of different things that can cause genetic changes in our cells. Some of these are things we've all heard about in preventing cancer. So increased exposure to the sun or UV and smoking can both cause damage in our cells and lead to these genetic changes. Sometimes though, even if you eat healthy, you exercise, wear your sunscreen, and you don't smoke, your cells can still get a genetic change, just through random chance and bad luck. In fact, every time your cell divides and splits in two, there's a small chance this can happen. But our cells normally have a great system in place to prevent this and fix it in the event that it does happen. So what does it mean to have a genetic change, and how can this lead to cancer? So if there's a genetic change in our cells, this means that something has changed in our DNA. And our DNA encodes for things called genes. It basically writes out the script or the word for each of these different genes. Our cells need this there to make the RNA. And RNA is kind of like the middleman or the intermediate step, because the cell then uses this RNA to make the proteins found in our cells. And these are what we think of as the workhorses in our cells. These proteins are responsible for carrying out all of the basic functions in the cell. They tell our cells when to start and stop dividing. They can detect these genetic changes and any damage in the cell, and then recruit other proteins to fix these mistakes. So any change to our DNA means that this code that our cells rely on to make the proteins is no longer right. There are many different types of genetic changes that can happen. You can suddenly have too many or too little copies of a gene. Or maybe the gene now makes too much of the RNA or protein. Or sometimes it's making the right amount of protein, but there's a change in the protein or a mutation that changes how it works, and it no longer works the way it should. No matter which type of change that happens, the result is the same. Our proteins no longer work the same, and this allows our cells to begin to grow uncontrollably and no longer responds to the cues it usually would to stop dividing. With so many different things that can happen in our cells, as researchers, we work together to solve these problems. As scientists, we work together with other scientists in many different fields, often outside of our own, to help unravel and solve this complex disease. And each discipline brings something unique to the table. They see the problem in a different way and can come up with unique solutions for these and help put a new piece together in the puzzle. By using this multidisciplinary approach, we're able to tackle the complexities of cancer research head-on. So getting to the stage of a new cancer treatment requires many years of work, not just in a clinical setting testing out these new treatments with patients, but also in the lab, using all of these different approaches from all kinds of different disciplines and bringing them together. So what I'm going to go through now is just one example of all the ways these different approaches can be used to try and develop new therapies for cancer treatment. So let's say a person is diagnosed with cancer and they're eligible for a clinical trial. So the patient will be diagnosed, they will be consented by clinical trials to take part in this trial, and then researchers will get a sample of that patient's particular cancer tissue. Now the patient would then receive treatment, which may be the standard of care that they would normally receive for that particular type of cancer, or maybe it's a new therapy that's being tested. Whatever the type of treatment is, the researchers would then also be able to gather survival data about the patients enrolled in the study. Now this tissue that we collect during the clinical trials is invaluable. From one piece of tissue, we can do genetic studies and search for those genetic changes that may have caused the cancer in the first place and may be causing it to progress or not respond to treatment. We can also take this tumor tissue and isolate different cells from the tumor. These cells can then be studied individually in a dish where we can monitor how they grow and respond to different therapies. And we can even take this a step further and inject those cells into little zebrafish embryos. These zebrafish are a great tool for studying new treatments in a quick and high throughput manner. And these cells or even pieces of the tumor itself can also be placed into specialized mice where the patient's exact tumor will grow. This allows us to get even more samples to study. Finally, we can use this tumor and use it for histology, which means we're looking at it under the microscope and we can stain it for different proteins, looking for changes in expression, and looking to see how the structure of that tissue has changed. As you can imagine though, finding a single genetic change within a tumor is like looking for a needle in a haystack. And this is where computer science can come in. We can take that tumor sample and isolate all of the DNA and get the full DNA sequence of that particular tumor. Once we have this sequence, computer scientists can compare this sequence of DNA to our normal DNA sequence found in cells. By doing this, they are able to more rapidly find that needle in a haystack and identify any unique genetic changes to that patient's tumor sample. This information can then be correlated with the patient's survival data, allowing researchers to potentially identify genes that may be useful for predicting patient outcomes or even developing new therapies. So let's say, for instance, gene X has been identified as having a genetic change in that patient's tumor sample. It could be that this means we have different amounts of that protein, or maybe that protein has been mutated. Either way, by getting this information from the computer scientists, biologists can now use our cell-based and animal models like the zebrafish and mice, and we can turn on or turn off this particular gene in these cells or in that tumor, and we can even make the exact same mutation that the patient had. When we manipulate or change the gene, we're able to ask, does this cause the cells or tumor to grow faster or slower? If we make this particular change in normal cells, do these cells now get characteristics of cancerous cells? And does changing this gene make the cells more resistant or more responsive to different treatments? All of this together helps us to better understand how cancer starts and how we can stop it. And importantly, by combining all of these different approaches together, we're able to much more quickly bring this back to the clinic to help patients. So our computer scientists identified gene X as a mutated gene, and our studies in the lab show that targeting gene X actually slowed the growth of cancer cells or even caused them to die off. Then maybe we can use this information to develop new therapies. Then we turn to chemists who have the tools to make these new therapies like inhibitors against gene X. And we could potentially develop a new therapy that would directly target this genetic change. Then, when patients come into the clinic and are diagnosed with cancer, if we're able to sequence through their cancer's tumor, we could then identify patients that have a mutation in gene X at the time of diagnosis. Taking these patients that have a specific mutation in gene X, we could then treat just those patients specifically with this new therapy. The hope is that this would increase patient survival and lead to better success in the treatment of cancer. By working together, we're able to gain new knowledge about cancer and new treatments faster than ever before. And while it still takes time to develop new therapies that will benefit patients, by working as a team, we get closer and closer every day.